can carry on in the...
Okay, I should not cut the call.
Yes.
Okay, my microphone ante phone lo vinipistundi, laptop lo vinipiyatledha.
Yes, ma'am. Yes, ma'am.
Okay, here turn up volume is there. Can I see once? ఇప్పుడు వినిపిస్తుంది ఏమన్నా?
Start presenting fine.
ఓకే, మొబైల్ కట్ చేయకుండా అట్లనే స్టార్ట్ చేస్తాను.
అట్లా వదిలేసేయండి, మీరు స్టార్ట్ చేసుకున్న ప్రెజెంటేషన్. ఎవ్రీథింగ్ ఇస్ ఫైన్.
ఓకే, మొబైల్ ఇక్కడ పెట్టేసి నేను స్టార్ట్ చేస్తాను. ఓకే, కెన్ ఐ స్టార్ట్?
Yes.
Okay, good evening, students. Myself, Alice, faculty of microbiology. So today's session is examination preparation session. As you know, in your subject, you have the microbiology and pathology, part A and part B will be there. So part A will be the pathology, which carries the 40 marks. Microbiology, part B, which also carries the 40 marks. Total paper will be the 80 marks. So you are writing two subjects, pathology and microbiology, 80 marks. Time limit will be 3 hours. So coming to the part A and part B, so the microbiology, and here if you see the question paper pattern, you may have the one essay that carries the 10 marks. You have the options in one A and B part will be there. So you may choose one question and you have to answer the one question. And coming to the next, in part B, only in microbiology, next 4 questions will be there. 4 into 5, 20 marks. And 10 plus 20, 30. So in the last 5 questions will be there. These are the 2 markers. So each question will carry the 2 marks. 5 questions into 5 into 2, 10 marks. So total yours will be, this carries 10 marks and 4 into 5, 20 marks. 30 and 5 questions, 5 into 2, 10 marks. So that is your question paper pattern. Time limit will be for 3 hours. So 1.5 hours you have to spend with pathology and 1.5 hours you can spend with microbiology. And coming to the tips, after completing the discussion of this question paper, I will let you know the tips for how to write to the exam, how you have to presentation and all. So coming to the first question here, A will be the, you can see the question, draw a neat labeled diagram of bacterial cell and describe the cell wall in detail. That is your question. So this may carry the 10 marks. They may split 5 into 5 plus 5. So coming to the neat labeled diagram of the bacterial cell or bacterial cell wall. So first you have to write about some points regarding the cell. Means what are the constituents, which are the components which are present in the normal cell. And you draw the neat diagram of the bacterial cell, which contains the ribosomes, mesosomes, Golgi apparatus, nucleus, and everything. Then what means meant by describe of the cell wall means in microbiology, you have to more stress about the gram-positive cell wall and gram-negative cell wall. Before writing that one, first you have to draw the diagram of the normal cell, bacterial cell with labeling. So the label also with diagrams always you draw with the pencil, and labeling also. So you put the arrow mark and do the labeling. So labeling you can do in all in one side. Okay, neat label diagram. And coming to the cell wall. So first, what is cell wall? What are the functions of the cell wall? So definition of the cell wall, main, what is the functions of the cell wall? What are the components? So first, peptidoglycan. So cell wall will give the rigidity to the bacteria. Rigidity means strongness. So you have to write points wise. So remember, don't write in the paragraphs wise. So always any question, use the flowcharts, or you may write in the points wise. For example, here cell wall. First, write down the functions of the cell wall. Rigidity upon the bacteria, and it will protect the cell wall and protect the cell content, cell membrane. And next, it prevents the osmotic damage. It gives the protection. And it also target site for the antibiotics. So name, first list out the functions of the cell wall. Then you can describe the cell wall. So cell wall may be differentiated gram-positive and gram-negative cell wall. So what are the contents which are present in the gram-positive cell wall? And what are the contents which are present in the gram-negative cell wall? Then what are the differences between the gram positive cell wall and gram negative cell wall. So main difference will be which cell wall will be thicker, gram positive cell wall will be thicker, because peptidoglycan layers are more than 80 nanometers will be present, which will be in the gram negative will be very less, 2 to 4 nanometers only. So that thickness which will be represented by the peptidoglycan layers. So in the positive cell wall, the peptidoglycan layers will be more. That's why the gram positive cell wall will be very thicker. So there you have to draw the diagram of gram positive cell wall, which contains the peptidoglycan layer, teichoic acids and all. Coming to the gram negative cell wall, structurally it is more complex. That means it contains the lipopolysaccharides, periplasmic space, peptidoglycan layers and all. But teichoic acids are absent in the gram negative cell wall. Main important in the gram negative cell wall is the lipopolysaccharide, which is responsible for the endotoxin activity that you have to mention. And outpouring membranes will be present. Outpouring proteins will be present. That so structurally gram negative cell wall will be complex, but thickness will be gram positive cell wall. So first you have to label the diagram of the bacterial cell, write down the points, label all the parts. Next, cell wall, gram positive cell wall, gram negative cell wall, gram positive cell wall. What are the contents regarding regarding gram positive cell wall diagram and gram negative cell wall diagram. Then you write down the major differences between the gram positive cell wall and gram negative cell wall. Next will be the demonstration. So what demonstration means, how we will demonstrate the cell wall. By seeing with our naked eye, we can't see that cell wall. So how we will demonstrate the cell wall? One is the differential staining is there, gram staining. So the gram staining is a differential staining which differentiates the whether it is a gram positive cell wall or gram negative cell wall. So the gram staining is a practical part. So we will have the 4 reagents in that one. After completing the staining, how we will tell that the this is a gram positive bacteria, this is a gram negative bacteria. So by seeing the color, if it is violet color, we can tell it is a gram positive. If it is a pink in color, it is a gram negative. Why all the gram positive will appear violet in color? Why the gram negative will appear pink in color? Why it is not taking the violet color? That is based on the theories of the gram staining. So major first is the cell wall. As I told you earlier, so cell wall thickness will be more in the gram positive, not in the gram negative. So gram positive cell wall will be more thicker. Because of the thickness, already the holes are closed. So when the pores are closed, whatever we are pouring the stain, counter stain, that is not entering into the gram positive cell wall. Already in the gram staining, we are using decolorizing. So in the third step, that gram negative will be in the decolorized state. In the fourth step, we are pouring the counter stain. So these bacteria, gram negative bacteria will take up the carbonyl fission or safranin, which is in pink in color. They will appear the pink in color. So gram positive will be violet, gram negative will be pink because of the cell wall thickness. So that main important point you have to mention. And next will be the demonstration, gram staining, electron microscopy, microdissection, plasmolysis. Like you have to write the names enough. No need of description. Then after demonstration, you have to write the cell wall deficient forms. 1 or 2 points enough. Name the cell wall deficient forms like mycoplasma, ureaplasma, L forms. So what are the terms? Protoplast, spiroplast. That's it, enough. So again, I am repeating for the answer for this question. First, label diagram of the bacterial cell. So you have to draw the diagram of the bacterial cell and label it and describe the cell wall means first cell wall functions, gram positive cell wall, gram negative cell wall diagram. So here completely you have to draw the 3 diagrams. Then cell wall components and gram negative cell wall components, main users, next demonstration and cell wall deficient forms. I think you clear this one, understand. And next coming to the R, that means either this question or this question you have to write. So what is the second question? Describe the laboratory diagnosis of pulmonary tuberculosis. So what my suggestion is, so first read the questions very carefully. So once or twice, first you read the questions. So then you understand what they asked. So don't write irrelevant matter to that. So and see that question marks and what are the important points should do. Write to that. For example, here the question is, describe the laboratory diagnosis of pulmonary tuberculosis. So where you read this one, which bacteria? Mycobacterium tuberculosis. So what they asked in the Mycobacterium tuberculosis, only the part of laboratory diagnosis. So you no need of writing all that morphology, culture, pathogenesis and all. You have to start with the laboratory diagnosis. In pathology, in microbiology, the laboratory diagnosis of all the bacteria will be important. Some of the bacteria you may read in your pathology also, but there the pathogenesis will be more important. Coming to the microbiology, laboratory diagnosis will be important. So any laboratory diagnosis of any bacteria, you have to remember these highlightings. So first you have to start with the specimen collection. So depends on the disease. So what are the specimens we have to take? And after that, direct microscopy. That means before inoculating into the culture media, direct microscopy. And then culture. So inoculate on the culture plates. After culture, then we have to go for the biochemical reactions to know the species level. And then if any other enzyme test. And then we have to go for the prophylaxis till treatment. So this is the order for laboratory diagnosis. Specimen collection, if any transport media is needed, we have to mention direct microscopy, culture, and then if any prophylaxis and treatment. So first we will see here laboratory diagnosis of the pulmonary tuberculosis. So tuberculosis is caused by the Mycobacterium tubercle bacilli or Mycobacterium tuberculosis. So it is a acid fast bacteria. So I think you know the staining. One staining I told you, gram staining, and that is a differential gram positive and gram negative bacteria. And one more staining is the Jaden staining. The name, the scientist, Gill and Nielsen. These both are the scientists who discovered the Jaden staining. So this also called as the acid fast bacteria staining. Why it is called as the acid fast bacteria staining? Because if you observe the cell wall of the Mycobacterium tuberculosis, so there one acid will be present, mycolic acid. Because of the presence of that mycolic acid, so these have the acid fastness. So in the Jaden staining, so 3 reagents will be there. So strong carbon fusion. In the decolorizer, we are using 20% H2SO4, which is as acid. So these bacteria, they are tolerating the 20% H2SO4. Still, they are appearing in the primary stain. That is the strong carbon fusion, which is a pink in color. So what is the sample we are taking in case of the tuberculosis? So first laboratory diagnosis means we have to start with the sample. Here you have to read the question carefully. Pulmonary tuberculosis. Why they mentioned pulmonary? Because TB, tuberculosis means it is not only for the lungs. TB may attack any organ in the body. But why only the lungs means these are the aerobic bacteria more favor to grow in the where oxygen will be more. So in our body, the oxygen will be more in the lungs. So that's why in the tuberculosis, lungs will be first affecting. It does not mean tuberculosis only affecting the lungs. It affects any organ in the body. That's why they mentioned the laboratory diagnosis of only pulmonary tuberculosis. If other than the lungs, the TB will be there. For example, to the brain, meningeal TB, or kidneys, renal, or any other bones. So that we are calling it as extrapulmonary tuberculosis. So you should no need to write about the extrapulmonary. So the question is pulmonary tuberculosis. You have to write about pulmonary tuberculosis only. So first start with the specimens. So what are the specimens we have to collect for this pulmonary tuberculosis? So we have to take the specimen sputum. So sputum also, so how to collect the sputum? Because specimen collection will be very important. Specimen collection, storage and transport will be very important in the microbiology. So the specimen should be proper or inadequate or inappropriate specimens. If we collect, that will be difficult to the laboratory technician to proceed. So here the sputum sample we have to collect. Early morning sputum sample we have to collect preferably. And then not only the one sample, 2 spot sample. If the patient will come in the afternoon, so during that time, we may collect the spot sample. Again, we will give one sample container to him, ask him to get the early morning sputum when they will woke up. So if they have the bout of cough, immediately that should be split into the sterile container, let it get to the lab. So first specimen collection will be the sputum. So after then the sputum will be mixed with all saliva and epithelial cells and all everything. So that sputum should be concentration. So some concentration methods. Why these concentration methods will be needed? To homogenate the specimen or to decontaminate the specimen, to kill any other bacteria, overgrowth bacteria. Okay, so then we have to collect the sample. Then if it is like one method will be, you have to write here Petrov's method. So in that concentration methods, Petrov's method, if you mention, enough for this one. So specimen collection, next concentration methods, Petrov's method, N-acetyl N-glutamine, and next you can write the Jaden staining. So this is the gold standard method for the diagnosis of the tuberculosis. If you have seen any report of the TB, I think you all of you know that some reports like HIV, hepatitis, HCV, like so they will give reactive, non-reactive, positive, negative. Any layman, they can see that report, they will tell that. But here, if you have seen that tuberculosis report, they won't give positive, negative. So some grading will be there. So that grading you have to write here. So first we have to explain the Jaden staining. So then what is the procedure, how the bacilli will appear. So in that pink colored bacilli with the blue color background, blue color pus cells will be there. So then, so what about the other staining? Other than the Jaden staining, gram-positive staining will be there. That will be poor staining. So we are not going to do the gram staining. Jaden stain will be a grading you have to write. So what is that grading? If for example, per field, when you are seeing that microscope, that we are calling it as a one field. 1 to 9 bacteria per field. If you have seen, that is the 1 plus 100 fields. So 1 to 10 bacteria, that if you are seeing like this, if you have seen 10 bacteria or more than that clusters, so that may be the 4 plus. So the AFB, they can give 1 plus, 2 plus, 3 plus, 4 plus. So specimen collection, next will be the concentration methods, next Jaden staining method, then culture. What is the culture media for the tuberculosis to grow? LJ medium. Lowenstein, Jensen medium. Lowenstein and Jensen are the scientists who discovered this medium. This is an egg-based media. Beaten eggs acts as a solidifying agent. So you have to write about that media and how the colonies will grow, like cream, tough, buff, rough, like colony morphology you have to write. Any laboratory diagnosis, when you are mentioning the culture, so you have to write about the colony morphology. Okay, clear? Next. After that culture, LJ medium, some other molecular methods are there. No need of writing in detail about the molecular methods. Jaden stain and LJ medium, blood agar, that you can write. And other media like some Dubose medium, Sula sartans, these are the Middlebrook, agar-based media. So this you have to mention, but in detail about LJ medium, how it will be sterilized in speciation. After that, you write down that Bactec, LRTD, PCR, like that, some molecular techniques, names enough. No need of in detail about that one. Okay. So then after that, culture, next you have to write the prophylaxis and treatment. So in the treatment, what are the drugs? Like rifampicin, isoniazid, ethambutol. So first line drugs, second line drugs you have to mention. Prophylaxis, what is the vaccine for that one? BCG vaccine. So what are the main points you have to include in the vaccines? Not only the BCG, any vaccines when you are writing, first elaborate the term BCG, Bacillus calmette guarin. Okay. Next, you have to write the dosage. How much dosage? Route of administration, where it is giving, like parenteral or subcutaneous, intracutaneous, like that. Route of administration and dosage, how much? 1.5 ml, 2 ml, like that. And then duration. So, for example, if any virus vaccine will be given 6 months or 1 year, booster dose 5 years, like that. So that is the laboratory diagnosis of pulmonary tuberculosis. So here they didn't ask about the extra pulmonary, no need of writing about that one. If they will ask the question laboratory diagnosis of tuberculosis, then you have to mention the specimen collection, pulmonary, sputum, extra pulmonary, meningitis means CSF, like that. Okay. So these are the essays. So I am moving on to 5 markers. So 4 questions will be there. 4 into 5 marks. And don't think it and don't assume that if you write the more papers, for example, essay question, if you write the 3-4 papers, you will get the marks. For 5 marks, if you write the 2 papers, you will get the mark. It won't depend on the pages. It depends on the content. Okay, so if you write the relevant points, what are the important points? So that will be enough for writing. And next is the urinary tract infections. So urinary tract infections, here it is the etiological agents and laboratory diagnosis they asked. So all of you know UTI infection, etiological agents means causative agents. So here the causative agents will be mostly E. coli, Klebsiella, Serratia, Pseudomonas. These and all will come under the UTI, urinary organism causings. And laboratory diagnosis. So coming to the laboratory diagnosis, first the sample will be collected, urine sample. That is also midstream urine sample should be collected. After collecting the sample, immediately sent to the lab. If any delay occurs, the sample should be stored in the refrigerator except CSF sample. Because CSF sample, the organisms which causing the meningitis, they may die at the temperature. So first you will list out the etiological agents. Bacteria, viruses, fungi. If you write this like this one, it will be more better. And laboratory diagnosis, in fungi you may write the candida also. Laboratory diagnosis specimen, urine, culture will be McConkey agar to differentiate the lactose fermenter and non-lactose fermenter. And next you may write the blood agar, because Enterococcus is there, Serratia is there. Some of the Staphylococcus, epidermidis are saprophytic. Normally they are the common only, but normally they are the non-pathogenic only. But when in the immunocompromised patient, it may become positive. And in the laboratory diagnosis, what is significant bacteriuria? That will be main important. So if you have seen any culture and sensitivity report outside also, for urine culture, they will mention like E. coli has grown after 48 hours. Beside that E. coli, they will give some value, 10 to the power of 5, 10 to the power of 6. That will be per CFU. CFU means colony forming unit. So like that. So what is that significance means? So when we are stick on the culture plate, Petri dish, so if the complete full growth will come, that will be considered as a 10 to the power of 6. So in case of 10 to the power of 2 or 10 to the power of 4, that we can consider it as an insignificant bacteriuria, presence of the bacteria in the urine. If it is more than 10 to the power of 6, we can consider it as a significant bacteriuria. So in the laboratory diagnosis, and one more thing important, any sample before collecting, ask the patient if they are on the antibiotics. So we should avoid if they are on the antibiotics, because nowadays the people also, they are without consulting the doctor, if they have any infection, easily they are going to the medical shops and they are getting the medicine. So after not subsiding, then they will come to the doctor. So you have to ask that. So before, so then you have to mention that point also. Prior to the antibiotic taking only, they have to give the sample. So laboratory diagnosis, you have to include the urine sample. After that, direct microscopy, urine should be centrifuged. So that supernatant should be gram stained. If it indicates any pus cells, RBC, epithelial cells, indicates the presence of infection. Then culture it on the blood agar, McConkey agar, nutrient agar. So next day, we can see the growth. So after doing that gram staining, we come to know whether it is a gram positive, gram negative, that and all you have to mention. Laboratory diagnosis. So sometimes they may ask for the essay, morphology, culture, pathogenesis, laboratory diagnosis. No need of, again, so in the morphology culture, we will mention in the laboratory diagnosis, if you mention the side headings, enough. But if they will ask only laboratory diagnosis particularly, so you have to write elaborately. Okay. Then after that culture, then you have to write about the treatment. Antibiotics, norfloxacin, ofloxacin, that. So that is the urinary tract infection. Next, laboratory diagnosis of the leprosy. So leprosy is the very ancient disease which is caused by the Mycobacterium leprae only. And here we have to collect the sample, specimens, nasal sputum. Okay, so we have to collect the nasal mucosa and also lesions, skin lesions also. Mainly 4 samples we have to collect: forehead, buttocks, cheeks, and nasal septum. We have to collect the sample. And next, after collecting this one, this procedure, we are calling it as a slit skin smear. Okay, so we have to collect the sample and we have to do the Jaden staining. So they may appear like a cigar bundle. The diagram we have to draw. What are the specimens we have to collect? That procedure, slit skin procedure we have to write. And next, animal inoculation also will be there. Skin and nerve biopsy also we can. Here also same that in the Jaden staining, Mycobacterium tuberculosis, here also grading system will be there. So, and here in this culture media, no, don't write any culture media like blindly blood agar, McConkey agar, like that. So remembered leprosy, leprobacilli is a very ancient disease, but still the mechanism is not understood. It won't be cultivable in any artificial media. It will be cultivable only in the animals. That is also 9-banded armadillo that you have to mention. Okay, and treatment and prophylaxis, there is no complete vaccine for this lepre. So that BCG vaccine only, because of the same genus, so it will give some protection. But here it may be symptoms will be appear very late. Some people 2 to 3 years, 4 years like that. So lepromin test will be there. So that you have to write. And this test will be like type 4 hypersensitivity is come in this lepromin test. So to differentiate that grading system differentiate the tuberculoid leprosy and lepromatous leprosy. So these points you have to write. And coming to the laboratory diagnosis of cholera. So cholera is caused by Vibrio cholera. So what is the diagnosis of this cholera means? First, we have to write the specimens. So what are the specimens we have to take? So here in this one, diarrhea, massive diarrhea will be there. So watery stool. So that stool may be also not thick, watery stool, like a rice water. It may be appearing like a rice water stool, and rectal swab also should be preferable. Mostly the watery stool only. So after collecting the samples, then culture should be done, like selective media will be there, bile salt agar, TCBS, thiosulfate citrate, bile sucrose, and enrichment media, selenite F broth, alkaline peptone water, and if needed, any transport media also, like Venkatraman and Ramakrishnan media, that and all will be available. So specimen collection, then any transport media, direct microscopy, culture. Media you should write. How on the media, how the colonies will appear, like TCBS, yellow color colonies will be appear. Like that colony description you have to write. And then what is the treatment for that one? So must we have to treat the dehydration by giving the oral, like any water and electrolytes will be given, or any antibiotics, ciprofloxacin, and we can give cefixime like that. And then no prophylaxis will be, vaccines are available. So the vaccines may be also like live vaccines will be there, and killed vaccines also will be there. So in the laboratory diagnosis of leprosy also we have seen like DAPSO, and in the cholera also some of the antibiotics we can give. And what are hospital acquired infections? These are also called as the nosocomial infections. The name itself, it implies that hospital acquired. When the patient is admitted in the hospital with one disease, during that time, he don't have this disease. So after admitting in the hospital, he may have that other disease. That type of diseases, we are calling it as a hospital acquired infections or nosocomial infections. Main important, what is the source for this hospital acquired infections are the main is the surgical site infections, central lines, and catheters. Okay, so all these are the main important thing. So main organisms, first you have to list out the organisms. First you have to write the definition of hospital acquired infection. What are the diseases which are caused by this hospital acquired? What are the bacteria will affecting Staphylococcus aureus, Klebsiella? Main Klebsiella pneumoniae is a ventilated associated, that we are calling it as a VAP, ventilated associated pneumonia. Surgical site infection, central line catheters, central line tubes. So this bacteria, so those who are on the ventilator, so this Klebsiella will be the more in the ventilated patients, then they may cause the hospital acquired. And foodborne also, through the food also, they may have a hospital acquired. Environment also will have the hospital acquired infections. So you have to write the definition, what are the organisms will be causing, and next, what are the next diseases which will be causing, that and all you have to write elaborately. Hospital acquired infections, and how to treat the hospital acquired infections also, maintaining the aseptic conditions and following the sterilization, that also you can include. So coming to the 2 markers, first is the name 2 antifungal agents. So first read the question carefully. Antifungal agents means you have to write about the drugs. So what are the antifungal agents? So if you take the ketones and azoles, like that any group you can write down to. Amphotericin B is the, and nystatin, and also ketoconazole, itraconazole, that and all you may, any 2. So better you may write the 4 antifungal agents. So 2 markers are the main important questions which you gain the full marks. If you write the correct answer, Then if you write the 2 names of the antifungal, you will get the 2 marks. Next is the name 2 bacteria, each responsible for the diarrhea and dysentery. So here you can remember, see the word diarrhea and dysentery. So they ask 2 bacteria, better you may write the 4. So what are the main bacteria you have to write the for diarrhea? First is the salmonella and dysentery, shigella. So don't write that entamoeba histolytica like that. So that's why read question properly. Why? Because here they asked 2 bacteria. So if they will ask bacteria, mention the bacteria only. If they will ask organisms, then you may write the bacteria, fungi, parasites and all. So you may write the salmonella, shigella, E. coli, and staphylococcus also will come. And next will be the that's it. So any 4 bacteria you may write down. Salmonella, Vibrio cholera also you can write down for the diarrhea. And next, what are the list of complications in the falciparum malaria? So what is falciparum malaria? Where you will get this one? In the malaria. So here complications, symptoms will be different, complications will be different. So symptoms we may see in everyone. Those who got the disease, they may have the symptoms, fever, anorexia, malaise, discomfort, everything will be same. But complications we may see in someone. So falciparum malaria means the malaria caused by the 4 species are there. So among that one is the falciparum. So here the complications of this falciparum malaria, main is the pernicious anemia or cerebral meningitis and black water fever. Okay, so that you have to write the complications. Main 3 or 4 you may write down that, and also acute kidney injury also you may write. And name 2 chemical disinfectants. So what are the chemical disinfectants? First you can list out. You may read this one in the sterilization and disinfection. So in the disinfection, alcohols, these are the chemical methods, aldehydes, phenols, halogens, oxidizing agents like that. Among that, you can select any 2. For example, if you're writing the alcohols, put the heading of the alcohol and write down ethyl alcohol, isopropyl alcohol. If you want to write the aldehydes, formaldehyde, glutaraldehyde, like that. Phenols or halogens, anything. So then you write down the uses also. Mention the name of the disinfectant and use. Formaldehyde, which is used for the fumigation of the operation theaters. Ethyl alcohol, which is used for the disinfectants, like that. Okay. Next. Name 2, this one. Last question is the gonorrhea. So gonorrhea is the disease. So if they will ask like common, they are not mentioning here pathogenesis, laboratory diagnosis, not like that. They gave like a word gonorrhea. So then this time what you have to write? No, all the points you have to cut it and write down. So gonorrhea is caused by the Neisseria gonococcus. It is a gram-negative cocci which are arranged in the pairs, diplococci. So small heading, morphology 1 point. Culture. It may grow in the Thayer-Martin medium, modified Thayer-Martin medium, which contains the vancomycin, colistin, nystatin, trimethoprim. What are the colony growth? Enough. Because it's a 2 marker only. But they asked the gonorrhea. So 1 point for that one. Morphology, culture, pathogenesis. So how the pathogens, urethritis, cervicitis, balanitis, how it will be occur in the males and females. Male, like a flow chart. Okay. And next, laboratory diagnosis, one word, what are the specimen we can take? Urethral swabs. So then you have to mention that laboratory diagnosis, culture, and one point in that one, and diagnosis, treatment, and then prevention. So if they will, common question, if they will ask 2 marker, 4 marker, like gonorrhea, tetanus, tuberculosis, like that if they will ask. If they ask, mention about the, like, complications, like they mention laboratory diagnosis, pathogenesis, if you write that much, enough. Okay, I think you understand what are the points you should write for these questions. Now I will tell in general. So in general, what are the tips you have to follow? Main important will be the presentation. Okay, so coming to the presentation, so you have to follow writing. Main writing will be important, and question numbering. So for example, here they gave 1A, B. So if you are writing in the laboratory pulmonary tuberculosis, you have to write 1, round it off, and in that B, you have to mention clearly. So you don't think that that is the 1. So you are writing second question, pulmonary tuberculosis. No, in the 1 only you are writing the B part. Okay, so that question numbering will be very important. And don't write in the paragraphs. Make it like a charts. flow charts, and whatever the questions, first you are confident, whether it is a 2 marker or 4 markers or essay. So that you start first. If you have started 2 marker, complete it. All the 5 you can complete it. And then go for next session, 4 markers. And next essays. That is the important essay question. And don't think that essay question, if you write 4 or 5 pages, not like that. Content will be important. So main important scoring will be in the 2 markers and 4 markers. If you mention the 2 points in the 2 marker, they will give 2 out of 2. For this 10 marks, so if you writing everything also 10 out of 10, very less they will give. So maximum 8 or 8.5 like that. So if you know that answer in that essay, you write down that, and 4 markers more concentrated on the main important points. So don't be in the tense if they will ask 2 marker as a 4 marker, 4 marker as a essay, like that. Don't think like that. So if the 4 marker will ask for the 2 marker, what are the important points you have to write? You can write down. If the 2 marker, they may ask for the 4 marker, content will be important. You can add some more points, like that. So as an example, gonorrhea, that you may write for the 4 marker also, in detail about the culture and all. Okay, so that will be the main important points. And writing also clearly had mentioned. And one more advice is, suggestion is, don't leave the space. So after one question, some people, what they will do, so they will start one question, in middle they don't remember the points, so they leave some space and they will go for the next page. So if that space, if you are confident to fill in the next, means you can leave it and you can fill that space. Because that examiner who is correcting your papers, they may have some question, oh, something will be missed, like that. Don't give that chance. Okay, so after completing the question, make one line, so that indicates that your answer will be completed there. And question numbers will be very important. Attempt all the questions. Okay, so if you don't know the answer for any one question also, write relevant points. So if any chances are in the last minute, they may give some marks to that one. And time limit. So most of the students, they may give that complaint like that, we didn't have the time. So where you will ask the time? So first, when you are reading, and also practice like that only. For example, essay question. So 10 to 15 minutes maximum. 2 marker, mostly 2 to 3 minutes, like that. So last 3 hours means last 10 minutes you can keep for the again rechecking. So did you give all the numbers, and all the question numbers will be right, and you attempted all the questions or not. And main important points, if you are confident only, then you can underline it. Okay, if you are not confident, don't underline that, and don't that will be more so. And next will be attempt all the questions, time important, and margins also. If you want to do any rough work or anything, means you can go for the last. And mainly for in the microbiology diagrams. So wherever the diagrams are required, so the diagrams you have to draw perfectly with the labeling. Most you can use the pencils. So when you are drawing the morphology of the bacteria, you may use the violet color for the gram positive, pink color for the gram negative, like that. Okay, so first read the questions carefully, and whatever the required points will be there, that you have to write. And this is about the tips and this one. Okay, if you have any questions, you may ask. I think I may end this one.